your questions in the chat window. And we will get those Q&A questions answered at the end of the talk. Without further ado, I'm going to start introducing Thomas. And this is basically our EV characterization series that we are organizing at SNEV. And today we have Thomas from Austria, who's going to be talking about visualizing EVs using electron microscopy. Thomas comes from having an extensive background in visualizing EVs using the electron microscopy technique. He has published over 30 papers in this field and has been selected for multiple keynote sessions and has been a speaker to invited talks across the globe in different conferences here in the United States as well as in the Europe. So we are very fortunate to have Thomas here joining us. And Thomas, it's all yours. Take it away.
Perfect. Thank you very much for this very kind introduction. Let me share my screen with you. So I hope you can see this. Good afternoon from Vienna. Once again, thank you very much for having me, for the opportunity to tell you a little bit about electron microscopy. And what I'm planning to do in the next half hour or a little longer, is to tell you about different methods, different approaches we can use to visualize extracellular vesicles by electron microscopy. So I figured out in this audience, we probably don't need to introduce what EVs are and why they are important. And therefore, I wanted to just jump into the topic. So there's many different electron microscopy methods, very different instruments available that you can use to visualize electron microscopy. And just to start off, I wanted to show you just like a short gallery of how some of these images from very, very different sources. I think I have worked with EVs from human cell cultures, mouse, milk, some very exotic samples from many, many different samples. And of course, there's also many different methods you can use to visualize them. After this short overview, what I'm really hoping you will take home with you after this talk here is I want to give you kind of like the full picture, which methods are out there, which methods can you use to visualize EVs. I will especially go into the differences between conventional techniques and cryo-EM. I want to tell you a little bit about not only what the strength of the different approaches are, but also what are their limitations, what are possible pitfalls, artifacts, what you need to look out for. I want to go into a little bit some differences between 2D and 3-dimensional imaging, and want to end with basically telling you a little bit about what I would consider the gold standard now for visualizing electron microscopes. So if you're not familiar with electron microscope and our world, this here is basically an electron microscopy grid, slightly larger than in real time, in real life. So an electron microscope grid is basically the sample support. So what a glass slide is for someone using a light microscope is basically an EM grid for us. And in reality, these things are just like 3 millimeters in diameter. They typically have kind of like a metal mesh, and then they have a support film on top of this. And this can be a film of plastic, this can be a carbon film, and it can be continuous, or it can have little holes in it, in this little model here, and also in the slide that I'm showing you here. And this is basically where our sample goes onto, and that is what goes into the microscope in the end. So here is what would happen if you take a sample. This is, I have to admit, not an EV sample here, but a liposome sample. and you would shock freeze it and throw it in the microscope. This is pretty much real time, and I hope you can see we're looking at a frozen sample. These images were recorded at around -180 degrees. And I hope what you can see is that this sample is changing very, very rapidly. What you're seeing here is basically the sample being destroyed. Yeah? Electron microscopy is a destructive technique. Therefore, we cannot do things like live imaging or so. Basically, we're destroying our sample as we look at it. And these white, let me just change my pointer, just a second, please. And these white holes that you saw, the movie is now repeating. I'll point them out to you in a second. These white holes that you see right here and here and here poking up, these are actually real holes in the sample. So we have basically completely cut water molecules into oxygen and hydrogen, really split them. And we've really destroyed all of the sample, all of the water surrounding it here. And if we would continue this, the whole sample would be basically evaporated, destroyed, and there would only be one big hole left. So this means if we want to visualize a sample, for example, an EV sample in an electron microscope, we cannot just put it in there without any treatment. Actually, we have to do some extensive sample preparation before the sample will be able to survive inside the microscope. One reason is electron radiation is a harsh radiation. It will damage the sample. And the other reason is all electron microscopes operate with a vacuum system, and all biological samples are filled with water, and that unfortunately doesn't go well together. So that would mean if you introduce a biological sample without any preparation into an electron microscope, in the vacuum, these samples would literally explode and the water would evaporate in no time. Yeah. So what we're trying to do is, I have this example here, we're trying to look at grapes and trying to describe them as good as we can. But if we're not careful in our sample preparation, the water will go away, we will introduce some damage to it. So we might end up with something like this here, like dried resins, which of course look very, very different from what we originally wanted to investigate. Yeah? And there's basically two approaches we can use for sample preparation: conventional approaches and more modern cryo approaches. I will come to them in a second. And there's also two different types of electron microscopy technique that we can use. One of these techniques is called scanning electron microscopy. This is a technique in which we're basically looking at the surface of our sample. Yeah? And then the majority of my talk will be about transmission electron microscope. This is the second technique. Here we're using electrons that are much stronger, and they will basically not bounce off by the surface, but they will penetrate the sample. And by penetrating the sample, we can learn a little bit about how the sample looks inside. This is what we see in transmission electron microscopy. And I've also given you here some examples. This here is a scanning EM image of vesicles, what you would see here. And down here to your right, you have a typical transmission electron microscopy image of an EV. Good. So for scanning EM, just to show, I have to admit, this is not my big area of expertise. I've done a little bit of scanning EM, but not with vesicles. So here this is taken from a paper, and where they recorded different types of vesicles at different magnifications. And this is, of course, good enough to see you have vesicles, whether they're present or not. You might be able to roughly discuss their shape. and their size, of course, but anything that's inside is invisible with this technique. And typically one key aspect that we're trying to do is if we can visualize the double membrane. This is still considered kind of like the gold standard, or that would be your final proof that this is really a vesicle, what we're looking at, and not something else, or an extracellular vesicle, not something else. Yeah, and this is something you can't do in scanning EM. This is something you can do in transmission electron microscopy, and the rest of my talk will be about this technique. Yeah. So in terms of sample preparation method, I briefly mentioned there's basically two approaches. A classical approach, a traditional approach. This was first invented in the 1940s, and some of these techniques have not changed much. Yeah, so this is not a very new technique, but still used very, very widely. Yeah. Since the 1980s, a new approach emerged, and this is cryo-electron microscopy. And both of these approaches, there's many different kind of like techniques you can use. And also somewhere in between, we have so-called hybrid methods. So these are methods that use a little bit of cryo-EM, but also a little bit of classical or traditional EM approaches. And what I want to do is basically, I want to give you an example for each of these techniques and show, briefly explain to you how these methods work, and show you how some of the images would look like that you can obtain with these. So let's start with classical or conventional electron microscopy. Here, what we do is we use very aggressive chemicals to fix our samples. So maybe some of you have worked with glutaraldehyde or paraformaldehyde. This would be some of these fixatives. In electron microscopy, we use a very specialized component called osmium tetroxide a lot. Yeah? These are all very reactive chemicals, and what they will do, they will form crosslinks within your sample. And these crosslinks, you can think of them as kind of like a stabilizing network, a little bit similar to a spider web, that will make sure everything stays in place where it is. Because what we're planning to do next, after adding these chemicals for fixation, we're going to remove the water. And then on top of this, we're also adding some heavy metals. They give us, this is basically a staining, they give us a lot of contrast. Yeah. So one thing that's very important about this method is that the result, the images that you finally will get, what you're visualizing here is not the electron density of your sample. So we're not visualizing the electron density of our EVs, of the proteins, lipids, or whatever other molecules might be involved here. But what we're visualizing is a heavy metal coating that we basically put surrounding these vesicles. Yeah. The big advantages of this approach is it's very fast, it's very cheap, it's quite simple, you don't need any fancy equipment. So this is all very straightforward, but you're limited in the resolution that you get, and you also have a risk of artifacts, and I'll come to that in a few minutes. So probably the most commonly used conventional or traditional technique is negative staining. And here is how this works. So you can think of this a little bit as if you would take your hand and put it in a soft mold. Yeah, this is what you see here. And then what you're visualizing in the end is not your biological sample, not your hand, but you're visualizing the imprint it left behind. Yeah, and if you do this kind of like sloppy, hopefully it'll still be good enough to decide or to see, okay, I have here something with 5 fingers. If you do this more careful, you might be able to see, okay, there's a ring on this on this finger, or you might even be able to see the fingerprints here in this conserved in this mold. Yeah? So in negative staining, how does this work? So what we take, the method is super simple. So we would typically take something like 5 microliters of your EV solution and put a drop on one of these electron microscopy grids. Yeah, then we give it around a minute so that the EVs get a chance to adhere to the surface of this grid. Typically, we would use a carbon film for these EVs to adhere to. And then we're adding a drop of a heavy metal solution. And then we let the whole thing dry, and that's it. Yeah, so it's super quick, super fast. This can be done in less than 5 minutes. And the images, I'll show you some examples, have the big advantage that because we use these heavy metals, they work as basically staining agents. They give us a lot of contrast. Yeah, and here are a few examples of how, for example, EVs can look like if you use heavy metal staining for them. Yeah. Once again, what's very important here, what you're visualizing here is basically the imprint, the vesicles left behind in the moment that they were drying out in this heavy metal staining solution. That's what we're looking at here. And yeah, the advantages I just mentioned are it's super fast. Somebody who has a little bit experience in this technique can go from applying the sample to your grid to actually getting an image in an electron microscope in something like 10 minutes. Yeah, so it's super fast, but it also comes with some downsides, and I want to point one of them out. So if you would take a look at these two images here, I'm quite sure if you stare at them long enough, you could convince yourself, okay, I see some round blobs here, there could be some EVs here. And if I stare a little longer, I can also point to something that could be EVs here in this image on the right. But the truth is only this image here on the left is actually a real EV sample. This image here on the right, I took by just taking buffer, by just applying a drop of buffer to one of these EM grids and then adding my staining solution. And this is the big downside of electron microscopy. Yeah, it's a little bit like almost like coffee ground reading, because in the end, your sample dries out, and every solution, unless you have very pure water, every buffer, every other solution will have some salts, some soluble components in there, and they will eventually kind of like crystallize, fall out. And these are the structures that you see here. And it's actually not simple. And I have to admit, we are slowly shying away from using negative staining for vesicle characterization, because to be very honest, I can't really tell you if this is a vesicle or not. I've seen some images that are really beautiful where I think nobody would doubt that this is a vesicle. But in our hands, we've come to the conclusion that the method is just not reliable enough, that there's too much interpretation. It's just very difficult to say, is this a real EV or not? Yeah, so to sum up the pros and cons of this approach, as I said, it's fast, it's cheap, simple, and because we're using heavy metals, we have a wonderful contrast. Yeah, but I'm not very confident if I see a round blob somewhere. Is that a vesicle? Is that a dried out buffer, some buffer salts? Is that a contamination from the sample? The method is not good enough. We don't have high enough resolution to identify the double layer membrane, which would be a good indication that we really have vesicles here. And yeah, because you're drying out your sample in the end, we have a high risk of drying or staining artifacts, meaning we will see something that looks round and think it's a vesicle, but in reality, it's something different. Yeah. Good. This was a negative staining example for a conventional method. Now I want to show you an example of one of these hybrid methods. So this is basically using a little bit of conventional methods and also a little bit of cryo methods. And the technique I want to show you is called freeze fracturing, followed by replica shadowing. And here is how it works. So imagine you have two metal plates and you would put your EV sample in between them. the two metal plates are connected by a lever, and then you close it and you freeze the whole thing. And then in the frozen state, you bring this into a specialized machine, a freeze fracturing machine, and then you crack open these two metal places again. Unfortunately, we cannot decide or control where this cracking line, where this fracturing line is going through. But if we're lucky, this will go exactly at a very interesting moment, at an interesting position. So this could be, for example, just near the membrane, where we think some extracellular vesicles are about to being released or so. Yeah. And then you get basically these two halves of you have your metal plate connected by a lever, you crack it open. Now you have your sample in two halves. And then what we're doing, this part was basically, we just took the sample and froze it. This is basically pure cryo-EM. And now comes basically the conventional part, which makes it all together then a hybrid method. What we're doing now is we're applying heavy metal. In this time, we're doing this with shadowing. So this means we would use a heavy metal, typically platinum, and evaporate this on both of these halves. Yeah, and then technical detail, we put another layer on top of it, a carbon layer. And then what we can do now is we can take the sample out of the vacuum, we can warm it up. And what we're looking at now is not the biological sample anymore, but we're looking at this metal imprint that we have generated in this method, yeah, by depositing this heavy metal stain on top of our sample. This heavy metal imprint that we generated is called a replica. The nice thing is this replica will stay as is. You can warm up your sample. You can even add some SDS or so to digest it. But this replica will preserve the imprint of your biological sample. And this is then something you can look at. To be fair, I have to admit, these here are no vesicles. These here are, these are no extracellular vesicles. This is basically an emulsion we got from a collaborator who mixed oil and water phases. So a little bit like, you can think of this like a very big special liposome. And this method is especially suitable for very large samples, which might be a problem. So if you have samples that are a micron or even larger, then some of the other methods, negative stain and the cryo-EM methods I'm going to show you in a moment, can't deal with these large samples very well. But this hybrid method here, this freeze fracturing, this replica shadowing is ideal for these samples. Yeah, so it could be a small sample, a few below a micron or so, or it can be something gigantic, 10 microns or even larger. It works for all of them. Yeah. But again, here as well, you will not have the resolving power to see the double membrane. Good. So introducing cryo-EM. Since the 1980s, people found a very, very different way of doing EM. And in 2016, this method was also honored with the Chemistry Nobel Prize. So how does it work? So in cryo-EM, we're not using any heavy metal staining. We're not fixing our sample. We're not removing the water. Yeah? So we're just taking the sample and we're freezing it as fast as we can. And this minimizes any of this drying artifacts, staining artifacts I mentioned before. Yeah. But it comes also at a price because we don't have any heavy metals in there. We have super low contrast. Yeah, the signal to noise ratio in cryo-electron microscopy is horrible. Yeah, sometimes it's really difficult to even see where your sample is. But the big advantage, even though it's a very, very noisy image that we're generating, the electron density we're looking at is really from the biological sample itself. It's not the heavy metal coating surrounding it, but it's really from the proteins, the lipids, nucleic acid, whatever biomolecules are in your sample. This is what we're visualizing directly. And this gives us the high resolution power that we need, for example, to visualize also the double membrane of EVs. Yeah, and this is considered, as of today, this is considered the technique you can use to visualize your samples as close to the native state as possible today. And therefore, this has also been identified in the, for example, in the Journal of Extracellular Vesicles as kind of like the gold standard for visualizing EVs. So how does it work? There's a few different approaches how you can prepare samples for cryo-EM. The most common technique is plunge freezing or immersion freezing. And this could be some, there are basically safe-made equipment, what you see here. There are commercially available blotting machines that can do this for you. Or there's still the old school where people literally do it without any aid, just with a type of tweezer and filter paper. Yeah, so here are the steps how it works. So here we have a tweezer and you can test your eyesight. Here you see there's a tiny little grid. Yeah, so this is again one of these 3 millimeter grids. And what we're doing now is we apply a small droplet of our sample, typically 4 microliters or so. We apply this on here. And then you can see here, I just need to move something here out of the way. And then you can see here how the drop is sitting on this grid. And what we're doing now is we're basically taking a piece of filter paper and we're blotting most of the liquid away. This is what's happening here. The reason why we often use these blotting machines is because they can do this much more reproducible than a human can do this. Yeah. If you ever get a chance to work with this method, this will be the most inefficient way to prepare a sample that you ever use, I can promise you, because you will lose more than 99.98% of your preciously prepared sample. Yeah. So the large majority of this drop of this sample that you applied will just be sucked up in this filter paper and go straight into garbage. But the tiny little fraction that's left will form a super thin film on your grid. And this grid here has tiny holes in it. And you will get a super thin film, sometimes only 10, 20, 30 nanometers in thickness. You will get this thin film here on your grid. The film is super sensitive. It will evaporate in no time. This is why we have a climate chamber here where we can produce very high humidity. And then we shock freeze the grid immediately here in, for example, in very cold ethane at temperatures of minus 180 degrees or so. And what's happening under these conditions is you're shock freezing your grid. Meaning if I would put my cup into a freezer, at around 0 degrees Celsius, the water molecules would rearrange. They form this highly ordered ice crystal, and this takes up more space, and my poor little cup would probably break. And the same would happen to EVs and all other biological samples. This freezing here that we're using here in this plunge freezing approach is way faster. It happens in fractions, or it happens in a few milliseconds. And what this allows you to do, this is too fast for the water molecules to rearrange, meaning they're still frozen in this random orientation they have in the liquid phase. And what you get looks like a solid, but it still has all the properties of the physical properties of a liquid. This is referred to as a vitrified state or as a glass-like state. Yeah, and this is the sample we want for cryo-electron microscopy. Yeah, I mentioned this plunge freezing method is highly ineffective. You lose more than 99% of your sample. And I quickly wanted to show you 2 other possibilities that we could use. And this here is a commercial video by the producer of this instrument here called Vitrojet. And this is the so-called cryo-writer. The big advantage of this approach is it uses orders of magnitude less sample. Yeah. So if you have a very tricky sample where you only manage to produce a few microliters or so, then this here might be a possibility. The way how it works, it has a small metal pin that you will see in a second. This here is your grid. It's prepared in a certain way. That's not that important. And then now comes the important part. Here is your sample drop. And here's a tiny metal pin. You take a little bit of your sample and you write a little line on your grid. Yeah. So you print a little bit of your sample on a grid and then you shock freeze it. And that's it. Yeah, the big advantage here, you will need nano or even picoliters of a sample, but not the microliters you typically use for this plunge freezing approach I showed you before. Yeah, so this was the so-called cryo-writer as an alternative to plunge freezing. And another alternative there is, I wanted to show you, is called a sprayer. And we even have music with it, or I'm not sure if you can hear it. So this sprayer works very similar to an inkjet. And again, this is also a commercial video by the manufacturer. Take it with a grain of salt. The way how this method here works is you use a very special type of grid, a so-called self-wicking grid. This is basically a grid like this, but then you have kind of like thousands or billions of little needles on top of it. And if a droplet hits your grid, it will be nicely dispersed on your grid. Yeah. So here you have a small capillary where you soak up some of your sample. And now you're spraying your sample. And while spraying it, your grid is basically moving past it. This is what you will see now. Yeah, this is the self-wicking grid with these nanowires on top of it, with little needle, and it picks up automatically and then basically put next to the inkjet spraying device. And then it's basically just rushing through while your sample is sprayed on. And then you wait a little bit to allow this fine needle to basically suck up and move away your liquid until you have a very, very thin film. And then you're basically shock freezing it in liquid ethane, similar to what you do in plunge freezing. Yeah. So these are three different approaches how you can, the bigger advantage again of this ink or of this spraying device is you need orders of magnitude less sample. So if you have a difficult to prepare sample, that might be a good idea. Never mind which of these three approaches you use to prepare your cryo samples. The next steps are all the same. What you will do now is you will look at your sample. And contrary to negative stain, where we added this fixative, this heavy metals for protection, now we have a very precious native sample without any type of protection. And that means as soon as we basically turn on the electron beam, we're slowly but surely destroying the sample, as I showed you in the very beginning in this time-lapse video. Yeah, so therefore we work in a so-called low-dose mode. So this means we're going somewhere close to the region where we think we want to be, and we take some very short images with very little dose to orient ourselves. Then we go to one region where we do a little bit of fine tweaking. So this region is far enough away that when we shine the electron beam here on this region, we're not damaging our real region where the structures of interest are. And then finally, when everything is set up, we take one good shot at our region of interest as at our EVs. And that's hopefully the good image that will make it into the paper. If we missed it, no worries. This one region here is burned, but there is thousands and thousands more on one of the small 3 millimeter grids that we could use instead. Yeah. And so you start with, this here is one of these grids. You start with basically generating an overview then you slowly zoom in into one of these holes that we have here on these grids. Yeah, so one of these tiny little holes you can hopefully see here. And yeah, and then you do a little bit of fine tweaking, and then in the end you take your good image here. Yeah. And yeah, here's just a few examples of how some of these EVs can look like. In some cases, for example, here, this is actually a human sperm sample. And here you can take a look at your EVs without any sample preparation, without any concentrating necessary. You can just look at the pure sample and see already some of these EVs. For most other samples, of course, we need to do some purification assays before we're able to see our image. Compared to the images before, I hope you can see that the contrast is not that great. We have a very weak signal-to-noise ratio, but we have high resolution, meaning, for example, here we can clearly see the two layers, the double membrane of our vesicles, which is still considered, yeah, kind of like a gold standard for proving that this is really an EV that you're working with. Yeah. We always think of EVs of one of these round shape, well isolated EVs, but in reality, very often you get kind of like more diverse structures. They can have all different forms. You can have kind of like one EV inside another EV, or yeah, they could be even more complex than this. Good. So cryo-EM, I told you, allows you to really visualize your sample as close to the native state as possible. So what does that mean? Does that mean we have a perfect image here? Unfortunately, not quite. So in all images in cryo-EM, you see these little darker spots here, 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 or here. These are ice surface contaminations. You have a cold sample, and there are several steps involved transferring your sample from where you prepared it to the microscope where you want to image it. And a cold sample is like a magnet. So if there's any humidity in the outside air, it will condensate and freeze on your grid. And you will see this here. Yeah. We also have the problem, most purifications are, of course, we're not working with liposomes where we just add a few things together in a test tube, but many of our samples will come from very complex media to start with and have a very long and complicated purification. And therefore, there's no way around that we have lots of background in there from our sample. This could be soluble proteins, other components we weren't able to sort out, cell debris, you name it. Yeah. So therefore, our images look a lot more noisier than, for example, if you work with test tube samples like liposomes or so. And then, as I said, EVs are not always these singular round shape objects as we often think of them. Here's a good example where you have an EV in an EV in an EV, like 3 or 4 layers of complexity. And what's important to remember is the images we generate, never mind if in cryo-EM or in conventional methods, the typical image you generate in a transmission electron microscope is a so-called 2D projection image. So this means from this image here, all the 3D information is put into one layer, and I might have a good guess that this is really one EV inside another one, but it could also be one EV on top of another one. Yeah. From this single image here, I could not prove that this vesicle we see in here is actually inside the larger vesicle or would be on top or on the bottom of it. Yeah. And one thing, especially for larger EVs, that's also important, our sample thickness is, we're limited to about somewhere around 1 micrometer in terms of thickness. Thicker samples, we cannot get a transmission electron microscope beam through it. Yeah, and if our sample would be much thicker, there is a chance that when we produce it, for example, when we do this plunge freezing method, we could compress our sample. Yeah, these are all things we need to keep in mind. So this cryo-EM, this plunge freezing method, is not perfect. There's still a few things that can go wrong, or where we could introduce modifications to our sample. Yeah, so for example, in terms of sample preparation, so here there are some concerns if you use, for example, ultracentrifugation, if you use a lot of high G forces. Could that be, for example, the reason why we get these, could that be the reason that huge G forces that make one vesicle go inside another vesicle? Or did it look like this right from the start in our starting material? Is that how it was originally produced? Yeah. These are still open questions or things we need to keep in mind, where cryo-EM images are just limited. So speaking a little bit more about this 2D versus 3D imaging. So yeah, I mentioned the typical image you get from an electron microscope is a 2D projection image. And so you can think of this kind of like we have a 3D object here in the front, and then we shine a light on it and we look at the shadow. This would be the 2D projection image. And we might have a certain idea what we're looking at. But we might be right, but it could also be something completely different. Yeah. So this here is an artistic example from two artists, Tim Noble and Sue Webster. I quite like, which I think makes I like it. I like this example a lot to show how the 3D shape of what we think an object is could in fact be quite different. Yeah. And therefore, there's also other methods that we can use in electron microscopy, in cryo-electron microscopy, to really look at the 3D sample, at the real 3D structure. And this approach is called cryo-electron tomography. And here's a simplified version of how it works. So we have a 2D image here. And what you see down here is a 1D projection. And sorry, this is not working. Sorry. And what you see down here is this 1D projection. And if I now rotate my image around for 180 degrees and record lots of different projections here, with the sum of these projections, I can basically get the 2D information back. And the way how this works goes like this. You take one of these 1D projections, and the trick is to display it at precisely the angle where it was recorded. And then you just extend it from one dimension into the second one. So you basically just copy it. Yeah. And then if you now add a second projection on top of this, and again, in exactly the angle where it was recorded, and you keep on going and going and going, you can see we're able to slowly get the information back. Yeah. So here, this was an example going from 1D to 2D, but we use this in reality to go from a series of 2D images to really 3D information. The example I show you here is not, I unfortunately didn't have a vesicle sample with this. This is another biological sample that might produce vesicles. These are cilia and flagella. Yeah, the big advantage of these cryo-electron tomography approaches is we get really 3D data and information. And here we don't have a 2D projection image. Here we can really look at it in detail in different Z heights. So with this image here, or with this approach, we would be able, with the vesicle I showed you before, we would be able to absolutely say, this was really one vesicle inside another vesicle, or it was one vesicle on top or below another vesicle. Yeah. So, and one other approach I wanted to show you, what's slowly coming up, is to skip basically sample preparation steps. This will not work for all samples. If you have a very low concentration EV, there's no way you can visualize it directly. That would be the famous finding a needle in the haystack. Yeah? And cryo-EM is a bad method for screening large areas. In some samples, for example, here, the sperm sample, you might be lucky, because here, right from the start, you don't need any purification. You might be lucky that the vesicle concentration is so high, by just freezing it without any additional concentration effects, you might be able to see them. In most other cases, we might not have we might not be able to do this. But one approach I wanted to show to you, I think it hasn't been used yet in EV, but if we want to skip this sample concentration procedure by ultracentrifugation or size exclusion, one possibility that we could do is an approach called cryo-FIBSEM. Cryo-FIBSEM. This stands for cryo-focused ion beam milling scanning electron microscopy, and this is what you see here. So this is now with a cellular example. So these here are single cells, and in cryo-EM this would be way too thick. We can't look inside, but we can use this focused ion beam similar to a laser cutter and basically cut ourselves a little window into this very thick sample. And now this previously hidden region deep inside a cell would now be visible for us. Yeah, and then we could transfer this to a normal cryo-electron microscope and then study it in more detail. Yeah, so this could, for example, be used if you want to see vesicles, if you don't want to if you don't want to extract vesicles or isolate them, but if you, for example, wanted to see vesicles in the moment when they're excluded from a cell, or you want to study how are they formed, then this method here, this cryo-FIBSEM approach, would allow you to study even these thick samples and open up these new windows into cells that were previously way too thick to be visualized. Okay, I'm almost at the end. So a little comparison of, I showed you a little bit, I showed you negative staining as an example of a conventional method. I showed you freeze fracturing as an example of a hybrid method, and I showed you cryo-EM or plunge freezing as an example of these cryo methods. If we want to rank them in terms of how much work is it to get such an image, negative stain, we're looking at a few minutes. And cryo-EM, depending on how you're set up, this is definitely hours or maybe even days of work. Yeah, it's also way more expensive. It's way more effort. You need more specialized equipment. But we also see a strong increase in quality going from conventional methods via hybrid methods to cryo-EM methods. Yeah? And as a little summary, so what could you do if you want to visualize vesicles as close to the native state as possible? What's currently considered the gold standard to do this? That would be cryo-EM. Yeah, because this is just simply the method that allows you to look at your sample as close to the native state as possible today. You have a low chance of artifacts, so these staining and drying artifacts you can completely avoid. But keep in mind, there could be, if your sample is very thick, you might compress it. As I showed you before, we also we're also missing some 3D information. Yeah, so if you want to decide, is this really a vesicle in another vesicle or just on top of it, this is still limited. Yeah, sorry, that was a little bit confusing. To sum this up again, so cryo-EM is the gold standard for visualizing extracellular vesicles, because with this method you're really visualizing the density of your EVs directly. You're visualizing the density of your proteins, of your lipids, other biomolecules you have. You're not looking at a heavy metal stain that is surrounding this. You're really looking at your sample as close to the native as possible. This comes at a price. We will have a very bad signal signal to noise ratio because we're missing heavy metals. We have this low contrast. We might have high background. There's no way around this, occasionally ice contaminations. It's also important, these are all not quantitative methods. If you want to measure how many vesicles are in there, electron microscopy is not a good approach for this. Yeah, they might not be evenly distributed. I mentioned about we're missing 3D information. And for most of these approaches, you need a very high starting concentration. Yeah, we're talking about something like this here, which will be very difficult to get. And in some cases, this cryo writer or this cryo sprayer might help you if you can only produce very low amounts of your sample. Yeah, good. So this is how an idealized liposome sample would look like. A real life sample typically looks like this here. And as I mentioned now, I want to close by pointing out cryo-EM is not perfect. There's still a few things you need to keep in mind, but this is considered the gold standard, the best you can do if you want to visualize EVs right now. And with this, I'm at the end of my talk. Sorry, it took a little bit longer than I anticipated. I am more than happy to answer any questions and want to stop by acknowledging where I work at the EM facility in the Vienna Biocenter and give credit to our funding agencies and who helped with this presentation. Thank you very much.
Thank you so much, Thomas. So now we're just wanting to take any questions. So feel free to send your messages, your questions to the chat, or if you want to raise your hand or just unmute yourself.
Yeah, maybe I can start. I don't know how to raise the hand in the
oh yeah, no worries.
website. Hi, Thomas. Always really clear and super nice. Yeah, probably, as you know, I do a lot of negative stain on the vesicles. Unfortunately, not cryo. We are not. Anyway, I do a lot of it, and I have more than a question. I have a curiosity. If you have this experience, I always ask myself if there can be a difference. I mean, because you show that when you do negative stain, basically you put the vesicles on top of the grid and you wait a little bit, and then they will be there, and then you go to the microscope. Basically, what I do is the opposite. So I put the drop of the vesicles on the parafilm, and then I put the grids on. I also try to do the opposite as you do. I saw some difference, but not so many. So my question has been always if the vesicles will be on top of the drop, so it's better to put the grids on, or the other way around. What is your thinking?
Yeah, both works. So there's many different ways how you can do negative stain. I described, we take our grid, put a drop on top of it. I've also done it exactly like you described, where you have your parafilm drops and then put the grid on top of this. I would not expect tremendous differences. I think you can, depending on how long you allow it to incubate, this will.
Yeah, I usually leave them 5 minutes. You do less.
So, for example, we for some samples do this shorter, but typically longer incubation times should get you more of your vesicles on your grid. Unfortunately, it could also mean that more of the other things in your sample, the things that we don't want, what's going on in the background, this might also come down to your grid. But I think there's many different ways how to do it. I think that's totally fine. And I wanted to point you maybe here to this paper, because they have actually done a nice comparison where they looked at different samples, they followed different staining protocols, and they also asked different people, so which of these images here do you like best, or which one would you select? Because I think this is, you can see these here are basically all images from the state, or sorry, each line, each row here is from one and the same grid, and you can see there are huge differences. I should point out.
Yeah, no, I have exactly this experience from different kind of samples.
Yeah, and I should also point out, this is also true in cryo-EM. We see this all the time, that there are huge differences in terms of the concentration. You might look at one region of your grid and there's vesicle on top of vesicle, and then you might look at another region where it's emptiness, and every now and then you see a vesicle. And this is why I think it's important to keep in mind electron microscopy is great to visualize EVs, but it's not a good method to quantify this. Yeah, and I think this is true for negative staining as well as for cryo.
And another quick question that I have is about the fixation step, because when I started to work with that, the protocol that we used, I mean, after basically leaving the vesicles on the grids, we also used glutaraldehyde to fix the samples. Then when Johanna Hoag came in my lab, that you know her very well, she started to ask to me, like, why do you do this? It doesn't make any sense. Then I said, yeah, probably you're right. I don't know, we have done always like that. So what do you think about this? It's totally unusable, or it can change something?
We typically don't use it. So we use fixative for other approaches that we use typically for other samples. For EVs, our default method would be without glutaraldehyde or paraformaldehyde. We do it very, very simply, as I mentioned, where we literally take a drop of our sample and then a drop of a heavy metal staining solution. Typically, we use 2% urinal acetate.
Yeah, I do the same.
There's also phosphotungsten acid and many, many other stains. In our hands, urinal acetate seem to work nicely. On the other hand, I would not expect, I mean, what I could see is that with the glutaraldehyde, this might, I could see a higher risk of forming some aggregates. So if you see huge clumps of your sample, then maybe it might be worth leaving it out and just try it. How does the same sample look without?
No, I tried, to be honest, a little bit, and I didn't see a big difference with or without. So I was just wondering if you tried, if you have this experience.
Yeah, I think glutaraldehyde might be useful if you're working with very large samples, but if you have small vesicles, smaller than a micrometer or so, I would consider, I wouldn't be convinced that you need it, that it's necessary. I don't think it will hurt, but I'm not sure if you will need it.
Thank you. Thanks.
You're very welcome.
Well, thank you. And so the next question is from Lucia, and Lucia's asked, what are recommended buffers to use in conventional TEM for human EVs coming from plasma, serum, or saliva, so as to minimize the buffer's signal?
Sorry, I didn't quite get this. Let me take a look. Sorry, you were sorry, I was just distracted. Apologies.
That's okay. Do you want me to ask it again? That's fine. Yes, of course. So it's from Lucia, and they've asked, what are recommended buffers to use in conventional transmission EM for EVs coming from plasma, serum, or saliva?
Yeah, I have a very pragmatic approach to this. the simpler the better. So if there's a way how you can get your samples in tris, heapers, phosphate buffer or so, that's perfect. If your sample is not happy under these conditions and requires glycerol or certain additives or very specialized buffers, then do this. So the main priority is whatever keeps the sample happy, because we want to visualize it as close to the native state as possible and not something that was degenerated. It doesn't help us if we use a buffer which has a very low background, but we're only looking at destroyed samples. So if it's possible from the isolation procedure here, I would start with something very simple, tris, heapers, phosphate buffer. And if you feel like this is not working, you need additives, then whatever is necessary.
Great, thank you. And I think, is it Nadezhda has raised their hand?
Hello, thank you for the presentation. It was very interesting and very in-depth. I never worked with any electron microscopy, but I recently gave my samples to the outsource to GEM, and it was negative staining. So my question is, I've got some pictures very similar to yours on your negative staining pictures, and there are some particles which looks like normal EVs in shape like erythrocytes, and there are some particles which are black. There are some particles which are flat white. Could you please find your slide with negative staining?
Of course.
Maybe I can find mine.
Oh, sorry. This one here, can you see it?
Yes, yes.
Okay.
The right one, the right and upper. Yeah, this and closer to that.
Yep.
Very similar to what I've got. And if it would be plasma EVs, I would think first of all it's maybe lipoproteins, but my sample is just bacterial extracell vesicles, which comes from media, and there are no lipoproteins, no any other source particles. So I'm still questioning myself. What do you think about this?
Yeah, for me, I do not want to discredit the method. I just find it very difficult to interpret. If I look at a cryo-EM image and I can see a clear double membrane, I have no problem telling my collaborator, I am convinced this is an EV that we're looking at here. And in negative stain images, I just don't have this confidence. I could look at some of these things and I can tell you, yeah, it could be an EV. I could be convinced that could be one, but it could also be something else. And yeah, you see even on a single grid, there is this huge difference in concentration, but also the staining makes a huge difference. So in negative staining, we need just the right amount of basically stain to cover your sample. If it's too little or too much, you get other effects. You can sometimes get positive staining or no staining at all. And this is also the reason I would guess why on some of these regions you see some of these blobs seem darker, some of them seem brighter. That could also be differences in the size of the sample and also differences on how much stain stayed on the sample in these situations. Yeah, so therefore, we just recently had or it's still ongoing, a project with a collaborator where we exactly facing these questions. So the method is very good if you need quick feedback. So for example, if someone wants to find out, do I have EVs in there or not? or they're comparing two different purification methods, I think you could spend a day or more in cryo-EM to figure this out. But here, probably negative staining might be good enough to tell you we see more of this in this approach than in the other approach. But yeah, I think if you really want proof beyond any doubt that this is a vesicle and not something else, then I fear you might have to use cryo-EM.
Thank you.
Thank you so much. Yeah, I was really interested by the is it the VitroJet cryowriter that you talked about, because I was trying to do some cryo-EM for my PhD on some bacteria, and it was so hard to find even just a single bacterial cell because so much of it was being blotted off. So sounds very interesting.
Yeah, these other approaches, this cryowriter or cryosprayer, if you don't have a lot of sample, then I think they're very promising, but they're also quite expensive techniques, and only a fraction of your grid will be covered. Yeah, so with plunge freezing or negative stain, you typically have a whole grid to choose from which region you want to look at. And with these other approaches, there's typically only a few spots on your grid that are suitable. But yeah, it's a useful alternative that's out there. These instruments are still very new, so not too many facilities have them, but there are some some larger facilities. So for example, in Europe, there is the EBIC at Diamond, which is basically a big a huge EM facility, and they have some of these quite fancy sample preparation equipment, and the idea is to make this accessible. It's basically the same idea as national centers have that are now slowly popping up in, I think, many countries. And I think that would be some of these fancy sample preparation equipment that you might have in addition to the fancy microscope you need for this, that could help you with some of the sample preparation. Yeah.
Awesome, thank you. And I think Shipra's got her hand up as well.
Yes. Hi, Thomas. Nice talk. I had a quick question. I was wondering, is it possible to study the EV biogenesis using cryo-EM? Like, it's just, I was wondering, is that something, an avenue that you guys have pursued, or what possibilities exist?
Yes, I think this is possible. I think so far this has mostly been done with hybrid methods, and here with slightly different approaches. So for the plunge freezing approach I showed you will only work for thin samples, so for anything thinner than a micron or so. And most eukaryotic cells are, of course, much thicker than that. There's another approach, for example, high pressure freezing, that you can use to also freeze entire cells or even small tissue pieces. And what many people will do is they will use this high pressure freezing to freeze your sample, to get basically a cryopreserved sample. And then they will switch to conventional methods where you use fixatives, dehydration, staining to basically fix your cell, so that in the end you have this plastic embedded, this resin embedded cell. And then you can use an ultra microtome and cut thin slices. And in these slices, you can then start to look inside the cell. And here, hopefully, you would be able to see, for example, biogenesis of EVs, how they are developing. To do this in cryo is possible. I don't think anyone has done it so far. And this would then be exactly this, what I showed here at the very end, this cryo-FIBSEM approach. Yeah, so with this, you could imagine that this here would be your eukaryotic cell, and you would cut a thin slice somewhere just in the middle, and then basically analyze this little slice here in a cryo-TEM, or maybe even do, cryo-electron tomography on it to get the 3D structure, and then to study these structures. This approach is still quite new, and yeah, not quite straightforward, let's put it this way. Yeah, so it will be quite tricky to find the right moment in time. Yeah, if somebody offers you that as a PhD project, I would be very hesitant, because you might spend many years working on that. But these instruments are now getting better and better. There's just a new type of these cryo-FIBSEM instruments coming to the market, where you can automize this also better, where you have an autoloader, you can do high throughput. And so I think this will, right now, I think in the EV field, I did a quick PubMed search before the meeting. I couldn't find anything if anyone has used it, cryo-FIBSEM, to study EVs already. I think some people have just stumbled on EVs in their slices that they looked at, but I think it's not kind of like a standard method for EV visualization yet. But I could imagine, especially for projects that you just described, where you're interested in biogenesis or so, I think this is definitely a method to watch out for.
Can I just ask one thing more about this 3D tomography and 3D reconstruction? Probably I know your answer, but I tried. What do you think about the chemical fixation of the tissue samples, and then the tilt and the 3D reconstruction?
Yeah, if you're interested in the 3D structure, you don't have to do this in cryo. You're absolutely right. You can do this with conventional approaches as well. So this could be, just simple classical chemical fixation that you were referring to. You could also do this with this hybrid method, this high pressure freezing, followed by so-called free substitution, where you would also, in the end, have a resin-embedded sample that you can slice into pieces and look into it. If you want to get 3D information of your sample, you can do this as well. But here,
but you are not against the chemical fixation. This is what I want to know.
I'm very pragmatic with these things, so I think it always depends on which instrumentation is available and what's your scientific question. What's important to keep in mind is with this method, the resolution will again be slightly limited. So with pure classical chemical fixation, I think it will be almost impossible to see the double membrane. But with this hybrid approach, starting with high pressure freezing and then doing this free substitution, here, if all goes well, you might be able to see the double membrane. Depends on, again, what you're trying to do. If you want to prove to someone without any doubt that this is an extracellular vesicle, then chemical fixation might not be the method of choice. But if you're interested in studying biogenesis inside a cell of EVs, and this is the only method available, yes, absolutely, go ahead and do it. And if you do this combined with tomography, then of course you will get valuable 3D information from it. Yeah.
Thank you so much.
Thank you.
So I think that might be all we have time for today. I know there is one more question, but perhaps we could get some emails exchanged or something, if that's okay.
Absolutely, as you like. Yeah.
Perfect. So yeah, that was a fantastic talk. Very clear, very easy to understand, and very helpful for a lot of people, I'm sure, in a lot of different fields. So Thank you so much.
Oh, my pleasure, and thanks again for having me. It was great being
course.
here.
Thank you
Thank
so
you.
much, Thomas. And thank you everyone for joining as well. That was fantastic.
Thank you. Bye-bye.
Bye.